Welcome back to the online class. Our topic is female gametophyte, which is one of the pre-fertilization structure in flowering plants. As we know, the structure of female gametophyte comprises of pistil, megasporangium and embryo sac. So let's discuss all these three structures one by one. First, let's discuss what is pistil, which we also call as carpel. So pistil and carpel, these two words, they mean the same thing. Okay, remember this. Each pistil has three parts, that is stigma, style and ovary. First of all, let's see stigma. Stigma serves as a landing platform for pollen grains. It is receptive spot. So stigma is receptive in function because it receives pollen grains from the anther. Okay. Next is style. It is the connection between stigma and ovary. So this elongated structure, which is known as style, it connects the stigma and the ovary. Next is ovary. It is the basal swollen part of pistil which bears one or several ovules. So ovary is the basal portion. See in the diagram, the lower portion is the basal portion. So it is what? Swollen in structure and it bears what? One or more ovules. And now let's see about the structure of transverse section of an ovary. So inside the ovary, we will get the ovarian cavity, which is also known as locule, and inside the ovarian cavity, we will get the placenta, and from the placenta megasporangium will arise. So this megasporangium is also known as ovules. Here this is the image of a transverse section of an ovary. So see here, this outermost layer is the ovary wall. Yes, and next to the ovary wall inside it is the ovarian cavity, which is also known as locule, and inner to the locule, inside the locule we have the placenta. Okay, so this is the placenta, and from this placenta what is getting developed? It is the megasporangium, which we call it as ovule. Okay, so this is the structure of transverse section of ovary. These are again few more images of transverse section of ovary depending on the number of locule present in the ovary. Okay, so if there is only two locule, then we call it as bilocular, and if there is three locule, then we call it as trilocular, and if there is four locule, then we call it as tetralocular, and if there is five locule, then we call it as pentalocular. Okay, so this L, the alphabet L which is written, it represents locule. Okay, this is also an image of a pentalocular ovary. In different angiosperm plants, the number of ovules may be one or many. So for example, in plants like wheat, paddy, mango, the number of ovules is only one in an ovary. So because of that, we get only one seed from one single fruit of those plants. Okay, and in other plants like papaya, watermelon, pumpkin, lemon, orchid, etc. Okay, in those plants we will get many ovules in an ovary. Okay, so because of that we get many seed in one single fruit. So you can think of fruits like papaya, watermelon, pumpkin, jackfruit, etc., isn't it? So in those plants we get numerous seed in one single fruit. Now let's see about the structure of megasporangium and its development. As we know, the megasporangia, which we also call as ovule, it arises from the placenta. So this ovule or the megasporangia is attached to the placenta by a stalk called funicle. So you can see the diagram to know which part is known as funicle. Okay, so funicle means a stalk by which the placenta and the ovule remains attached to each other. The main parts of megasporangium or ovule are hilum, integuments, nucellus, micropyle, chalaza and embryo sac. As I keep on explaining, you can see the diagram as well. Okay, so hilum. Hilum is the junction between ovule and the funicle. Here junction means meeting point. Okay, so you can see the diagram which portion is known as hilum. Integuments. Each ovule has one or two protective envelopes called integuments which encircles the ovule except at the tip. There are two integument layers, that is outer integument and inner integument, as you can see in the diagram. So we will find the integuments around the ovule for the protection of the ovule, but at the tip portion the integument will be absent. Another part of an ovule is a nucellus. Integuments encloses a mass of cells called the nucellus, which have food reserves for female gametophyte. So inside the inner integument layer we will have the nucellus, and this nucellus is a mass of cells, that means it is a group of cells, and they have food reserve which is for the female gametophyte, that means for the developing embryo sac. So they will give nutrition to the embryo sac. Micropyle. It is a small opening present at the tip where integument is absent. Okay. Chalaza. It is the opposite to the micropylar and representing the basal part of the ovule. So chalaza, we will always find a chalaza at the opposite of the micropyle, and so it is the basal part of the ovule, and micropyle is the tip portion. Okay. The last part of the ovule is the embryo sac, which is also known as female gametophyte. It is located in the nucellus. So we will get the embryo sac in the nucellus, which is formed from a megaspore through reductional division. So we get the embryo sac from the megaspore by meiosis type of cell division, as we call the meiosis type of cell division as reductional division. So till now we have discussed about the main parts of the megasporangium or ovule. Now let's see what is megasporogenesis. The process of formation of megaspores from the megaspore mother cell is known as megasporogenesis. So here megasporogenesis means formation of megaspores. From where? From the megaspore mother cell. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. So we will get the megaspore mother cell at the micropylar end, okay, at the tip portion. The megaspore mother cell undergoes meiotic division and results in the production of 4 megaspores. This diagram shows the formation of megaspore tetrad from a single megaspore mother cell. Okay, how a single megaspore mother cell undergoes meiosis 1 and give rise to a megaspore dyad, and from the megaspore dyad again it undergoes meiosis 2 and give rise to a megaspore tetrad. So far I have explained you about the structure of pistil and megasporangium. So in our next video we shall continue with the development of embryo sac. So thank you.